not actually. Well, there is, but it's not actually going backward. It's like
Okay, thank you. Okay, good morning. So let's start so we can finish on time. So today, as you know, we'll be reviewing smooth muscle physiology. And here again is my information. Please contact me with any questions that you may have. Okay, so let's talk about the function of a smooth muscle. As I have it written here, there are many functional roles depending on the type of hollow organ that the smooth muscle lines, right? So then the goal of today's lecture is to understand the different types of a smooth muscle and their control and mechanisms for contraction. Okay, so as I said, there are different functions of the smooth muscle, and here I have some of them. That is that the smooth muscles maintains pressure, and I have this micrograph where you can distinguish all the smooth muscle cells. They are very tightly packed, and you can recognize that they have their spindle shape, right? So this arrangement is in a circular layer, and this is what will maintain the pressure against the contents of the organ. So in this case, we are looking at an arteriole. So another functional role is to mix and propel contents, and the cells, they will do that by adopting this configuration, a circular layer, but also they do have this longitudinal layers. And this is what will guarantee the peristalsis, right? Because you find this arrangement in the intestine wall, and then their activity will result in the mixed propulsion of the contents. Then another role is that they regulate the internal flow of the hollow organs. I'm talking about the bladder, rectum, the small testicular duct. And you see that now the smooth muscle cells, they are arranged in a rectangular layer. So this micrographs exemplifies how the diverse structures that the muscle cells could have, and also that means that will explain the different functions that the smooth muscles have. So in this cartoon, I have represented two smooth muscle cells. I guess that you are familiar with this because you review already this material. So just I want to remind you that the smooth muscle cells, they do contain this myosin thick filaments, and also contain this thin actin filaments. Also, as I have it here, you can appreciate, they are arranged in the same pattern that you find in the striated muscle. That is, in the striated muscle, remember, the myofilaments are arranged transversally. In this case, for the smooth muscle cells, you find organization more diagonally. So we have another filaments that are called intermediate filaments. They are not contractile elements, but instead they are part of the cytoskeleton. and they, of course, contain different type of proteins. They also have these structures that are called the dense body that are analogous to the C lines in the striated muscles, and that is because they anchor the thin filaments, as you can appreciate here, and also they do contain the same proteins that the C lines had in striated muscles. We have this the same type of structures, but now at the level of the sarcolemma, and we have what is called the dense area, and these are points of mechanical coupling. That is that these are points where the cells can pull on each other, right? So one of important or main difference between the smooth muscle and the striated muscle is that they don't contain troponin. Do you remember the role of troponin in striated muscles? Binds calcium, right? So in this case, you are guessing by now that still calcium is needed for contraction, as it is needed for in striated muscles. But now you can guess that the calcium will have to do it through a different mechanism, because the cells, they don't have, the smooth muscle cells, they don't have troponin. They also don't have the T-tubules that we describe for the striated muscle. And you can think that it's not necessary, because usually the diameter of the smooth muscle cells is small. So actually, if calcium needs from outside needs to reach the interior of the cells, it can do it in a fast way. So they don't have T-tubules. But they do contain the sarcoplasmic reticulum, because it's there where the calcium is being stored, as is the case for the striated muscle, but it's less developed. So then, as you see that we have the thick and thin filaments, so for contraction, the contraction will occur following the sliding filament mechanism. Remember that we described this mechanism for the striated muscles. And also, in addition, the contraction of these intermediate filaments is what it will pull the dense bodies together and will result in the shrinking of the cell. Okay, so another important structure is the presence of these gap junctions. Remember that cardiac muscle cells, they do have gap junctions, and this will also serve for points of functional coupling. So that means mainly because the electrical signals will be propagated through this type of junctions. So, so now we said that the smooth muscle cells contain gap junctions. If we take into account the presence of this degree of electrical coupling, we can classify the smooth muscle into a single unit smooth muscle or multi-unit smooth muscle. The features of a single unit is that is the most common. It's also referred as a visceral muscle. And as I said before, they are electrically coupled because they have many gap junctions. So then they will contract as a single unit. Right? There's something similar, let's say, that occurs in the cardiac cells, right? So then also these cells are self-excitable. That means that they will initiate an action potential. We'll see this type of activity later. Another feature is that they don't require nervous system stimulus to contract, and often they are modulated by ANS and other factors. In terms of the features of multi-unit smooth muscles, as I said, they have relatively few gap junctions. That means that they will contract as multiple discrete units, and they do require nervous system stimulation to contract. This is different compared to another difference compared to the single unit. So I want to make it clear that we already talked about this classification, and in the next slides we will talking about another types of classification. But all these classifications basically are done in order to understand or to describe the different properties of the smooth muscle cells. But you need to keep in mind that the classification is an oversimplification and then becomes difficult to separate in some tissues. Okay? Questions? No? Okay. So now, if we classify the smooth muscles accordingly of the type of contraction that they experience, then we can classify the muscles as a tonic muscles, and these are the ones that experience tonic contraction. That is that they will maintain a continuous level of partial contraction, as is illustrated here, and you find actually this type of activity in the cells that are found in the walls of the most blood vessels, and also in the airways or in the sphincters. So you see that there is a long maintained partial contraction, and this is what is referred as a tone. Everybody is familiar with tone, and also that's why it's referred as a tonic contraction. These contractions will result from when you apply a long duration stimulus, as is illustrated here. And as I said before, in order for contraction to occur, we need calcium, right? The calcium increases in the cytosol, as is illustrated here. The calcium rises, peaks, and then it will decline to a new low steady state level, as is illustrated here. And as we'll see later, in order for contraction to occur, we need the phosphorylation of the cross bridges. And here I have the temporal course of the cross bridge phosphorylation here in green. As you can see, basically follow the temporal course of calcium. It peaks and then declines to a new low steady state level. So the other type of contraction is the PHYSI contraction. Then you found this type of contraction in the muscles that contract rhythmically, like the GI, reproductive, and the urinary system. See, and then I have here the temporal course. Usually the PHYSI contraction, they are triggered by a small duration stimulation compared to the tonic contraction, and see that because with this stimulation you see an increase of intracellular calcium that peaks, but this one, it will go back to after some time will go back to the basal level. Then when we look the time course for the cross-bridge phosphorylation, it follows the same as the calcium, right? It peaks and then later goes back slowly to the basal level. And then here we have the resulting contraction, right? This is more like we will expect for the striated muscle, right? One contraction. And you can see this is quite different, the temporal course between these two types of contractions. And another difference is that this type of contraction is most of the time, if not always, initiated by an action potential. So an action potential would generate the PHYSI contraction, and if not, an appearance of an action potential will increase the contractile response in the PHYSI contraction. But in the case for tonic contraction, the muscles that experience this type of contraction, there is no need for action potential to initiate the contraction, although the changes in voltage membrane, they will affect the amount of force. And also, as is illustrated here, this contraction can occur at low levels of calcium, right? See? It's clear the difference between these two types, right? Especially by having in mind the temporal course of those events. So now we said that we need calcium for contraction, right? As is the case for striated muscles. So here I have a cartoon. You don't have it in your notes, but you already know this material, right? So in the case no, and I think I have it. I'm sorry. I have this in your notes. So this cartoon just represents all the sources that will contribute to elevate calcium in the cytosol. So here we have that this represents the voltage-gated calcium channels. This basically the same different isoform, but the same that we have in the striated muscles. So they will allow the influx of calcium, and this calcium will diffuse and reach the ryanodine receptor, activating through a mechanism that is calcium-induced calcium release. It's a mechanism similar that we described for the cardiac cells, right? So then this will respond releasing calcium. Here I have another group of channels that either could be ligand receptor gated or stretch activated calcium channels that they will also at some point allow the influx of calcium and that calcium will use for contraction. Also, as illustrated here, another mechanism that it says that when you have an agonist that activates some membrane receptors, and if this receptor is coupled to a G protein, it will lead to the production of this second messenger, the inositol triphosphate, and this will result in the activation of the inositol triphosphate receptors, the IP3 receptors, and the activation will will release calcium into the cytosol. So these are the sources of calcium for contraction in the smooth muscle cell. Also here, as you can see, we have this kind of small invaginations that are known as caveoli. That seems like in these regions is where you find the channels that will allow the entrance of calcium. So then we know that calcium is needed for contraction, as I said before, but it won't bind to troponin because the smooth muscle cells, they lack troponin. Instead, it will bind to calmodulin, and this is illustrated in this cartoon. So here I have that once calcium in cytoplasmic calcium rise in the cytosol, this will come and bind to calmodulin, and then calmodulin will activate the myosin light chain kinase, as is illustrated here. Then this kinase, in turn, will phosphorylate the myosin light chains in the head of the myosin, as is illustrated here, and this phosphorylation will favor the attachment or the interaction between the cross bridge and the thin filaments, as is illustrated here. And after this occurs, then we'll go into the cross bridge cycling. That is the same steps that we described for the striated muscle, right? Everybody remembers all the steps involved in the cross bridge cycling. So then one of the differences is that this cycling, the kinetics is much slower in the smooth muscle cells. And you have to think that usually the contraction for the striated muscles occurs in 1 second, let's say, but for the for a smooth muscle cell, the contraction could last several seconds. So the cross bridge is lower. So then you see that the rate limiting step for contraction is the phosphorylation of the cross bridges, as is illustrated here. So then we can say that the calcium switch for contraction in a smooth muscle is calcium-dependent phosphorylation of the myosin light chain. And this is different than what we described, right, for the striated muscle, that we said that the calcium switch is the calcium binding to troponin, right? Questions? No? Okay, so now let's talk about relaxation. We talk about contraction, how relaxation occurs, and usually this is through the same mechanism that we observed in striated muscle. What I'm talking about is the increase in cytosolic calcium needs to go back to the resting levels, right? And this is accomplished, as is in the case for the striated muscles. In the plasma membrane, we have the calcium ATPase and the sodium calcium exchanger. And at the level of the SR, we have this calcium ATPase, right, the CERCA, that will bring back the calcium inside of the SR. So when the cell lowers the cytoplasmic calcium concentration, then this will result in the activation of the myosin light chain phosphatase. When this is activated, this will unphosphorylate the myosin, and this will keep the head of the myosin close to the thick filaments. And this will result into relaxation of the smooth muscle. Okay? We have another mechanism that you already reviewed in the previous lecture, and that is if you have activation of the PKG or the PKA, this will come and phosphorylate the myosin light chain kinase, and this became inactive this kinase, and this will result in relaxation. So here we have 3 different mechanisms that will result in relaxation of the smooth muscle. Questions? No? Okay. So now let's move and talk about the mechanical aspects of smooth muscle contractions. You already recognized what I have here is the cross-bridge cycling that, and it's very similar that what you review in striated muscles. So the difference, and it's illustrated here, is the high probability that the myosin light change in the myosin is phosphorylated, as is illustrated here. So if the phosphorylation occurs, then we go to this step, and then, as I said before, then it will result in the attachment of the myofilaments. And because the head of the myosin is energized, why the head of the myosin is energized in this step? Because what? Is phosphorylated as is illustrated here, but the phosphorylation is in the myosin light chain kinase. But see that I have the cross bridge in red before that the phosphorylation occurs. So why this is showing in red? What do you need to occur in order for the myofilaments to detach? ATP needs to bind to the myosin, and once the ATP binds, the myofilaments detach, and the ATP is hydrolyzed. Remember that the head of the myosin has this intrinsic ATPase capability. So in this cartoon, is representing the head of the myosin is already energized, and then the myosin light chain is phosphorylated. So then we'll go to this step, and the phosphorylation allows the attachment of the myofilaments, and because the head of the myosin is energized, it will do this. How do we call this? The power stroke, right? Remember that is the responsible of the sliding of the thin filaments over the thick filaments. So then we'll go to the power stroke, and then here again we need one molecule of ATP to attach to the head of the myosin in order for the myofilaments to detach, right? So now you remember the whole cycle. So this will continue as long as phosphorylation is occurring, and this is because calcium level is high. And then this cycle will stop when 100% of the cross bridges are unphosphorylated. That means that because there was a decrease in the calcium concentration, right? So now you can put everything in context, I guess, because of this cartoon. So we are talking about here as a fast cycle. But as I said before, you have high probabilities that also that the myosin gets unphosphorylated, right? And when this gets unphosphorylated, let's see what it happens. For instance, if the probability of a cross bridge is 75% to get unphosphorylated. When this occurs, then the cell will go through this slow mode cycling that is illustrated here in black. So the difference between these two cycles is that this cycle is a slow mode. That means that there is a reduction in the cycling rate of the cross bridging. Okay, so let's say here. So here the myosin was phosphorylated, but then it got unphosphorylated, as is illustrated here, because the myofilaments were attached. So then they are going to be able to produce the power stroke, and then we'll continue the cycle. But this transition will occur way more slower than occurs in the fast cycle. And is illustrated here. In each one of these steps, you always have the possibility that the myosin light chains can be phosphorylated and enter into the fast cycle. See? Then we see that when the smooth muscle enters into this slow mode is what is known as a latch state. And latch state is the slow cycling of the attached cross-bridging that maintains the sustained force. And what is the advantage of this type of system? You guess by now, right, that the advantage is that the response to a sustained stimuli will be maintained without further utilization of ATP. So we are being economical here, right? So, and this is what occurs. Remember when we described the tonic contraction, we see that the cell can maintain for long periods of time this type of contraction, even though that you have low levels of calcium and then low levels of phosphorylation. So that means that here, mostly after it goes to a fast cycle, it will transit to a slow mode cycle. And those mechanisms are the responsible of this maintained response. Did that make sense? Yes? Okay. So now let's talk about the different mechanisms of producing contraction, because you can guess that the cells having so many different roles of function, it should have different mechanisms of contraction. So then we will talking and we'll see that the cells has different patterns of excitation contraction coupling. So here I have just listed some of the factors that will either induce or modulate contraction. I'm talking about the electrical activity, hormones, autonomic nerves, and drugs. So we'll see how all these different factors will end in contraction. So, but we have to keep in mind, right, that like a skeletal or cardiac muscle, contraction of a smooth muscle is dependent on calcium. So that means that all these factors that I just listed will induce contraction by increasing intracellular calcium concentration. Okay, so now let's talk about the electrical activity. That is one of the factors that can induce or modulate contraction. So when we look at the electrical activity, we found some smooth muscle cells that they are self-excitable. That is that they experience pacemaker potentials or slow wave potentials. In the case of the pacemaker potentials, this will occur as is represented in this cartoon. Usually the resting membrane potential in smooth muscle cells is higher, right, than striated muscle. Usually it's between -70, -50 millivolts. So then by the activity of some small cationic current, you see that the membrane potential depolarizes and then reach the threshold and then trigger an action potential. So then the cell repolarizes, but because of the activity of this current, it will take again, raise the membrane potential until we trigger another action potential. So this type of activity is very similar than the activity that you review in your heart lectures, right? For the SA node or AB node activity, right? The pacemaker cells. So this is a similar activity that you found in the smooth muscle cells. For the case the slow wave potentials, then the cell will experience these swings of depolarization, hyperpolarization, normally due to the activity of electrogenic pumps. And then when they are challenged by a particular excitatory stimulus, you see that now these oscillations are of greater magnitude. And then at some point they will reach the threshold, and you see what happened. It will trigger action potentials. Usually it will trigger bursts of action potentials. So then this activity is one of the different mechanisms that the smooth muscle cell uses to produce contraction. So here what I have is a relationship between the membrane potential and the generation of force here in the bottom. As you can see, the activity that I just described, this swing in membrane potentials with the appearance of action potentials on top of these swings, you see how if the burst contains many action potentials, this will be transduced in a stronger force of contraction, as is illustrated here. And you observe usually this activity in cells that are in the GI tract. So of course, because they experience this rhythmic activity. Here I have exemplified another mechanism for contraction. This is the most typical that we are familiar, because it's similar what occurred in the striated muscles. So here we have an action potential with the corresponding contraction or twitch. And look what happened. If we increase the frequency of these action potentials, we observe this phenomenon known as summation. Remember that we described that the skeletal muscle used this mechanism in order to graduate the force of contraction. So the smooth muscle cells also uses summation, even can go into tetanus to increase the force of contraction. So then I have another mechanism that consists in slow changes in the membrane potential. As you can see here, chronic hyperpolarization and depolarization. And the corresponding, you see relaxation, contraction. And actually, this is the mechanism that is responsible for the vascular tone. Remember the tonic contractions? So here you see how they occur. So, and we have another mechanism that doesn't involve significant changes in the membrane potential, as is illustrated here, but still you can obtain the cells to contract or to relax while they are under the influence of some hormones, neurotransmitters, or drugs, right? So this type of mechanism is what is known pharmacomechanical coupling. In the other three mechanisms is the type of excitation contraction coupling. In this case is pharmacomechanical coupling. Questions? Okay. So as I said, another factor that influences contraction, that influences tension generation, is the innervation. And then I think that probably you already reviewed this in your previous lecture, in your ANS lecture. And actually the innervation, what it does, it will modify the rate of activity and the strength of contraction. That would be the way of regulation. And one important one feature is that the axons of the ANS do not synapse with the smooth muscle cells the way that they do in the striated muscles. I mean, the striated muscles, remember that we have these specialized neuromuscular junctions, For the case of the smooth muscle cells, the axons will travel across the surface of the smooth muscle cells, as is illustrated in this micrograph. You can distinguish here the axon, and this axon contains these periodic bulges that are known as varicosities. And it's in here in these varicosities that contains and will release the synaptic vesicles that contains the neurotransmitter, right? Then the neurotransmitter has to diffuse in order to reach the adrenergic or cholinergic receptors that are dispersed over the entire surface of the smooth muscle cells. Okay? So I have here that this pattern of innervation is very common in the multi-unit smooth muscle. See? And you can see in this cartoon that because we mentioned that one of the features of the multi-unit is that they don't have gap junctions. So that means that each cells, in order to contract, needs to be under the influence of the innervation, as is illustrated here. We have the varicosities. They release the neurotransmitters that diffuses and directly will activate the cells, and maybe they can reach for neighboring cells. But basically, each cell needs the input of this varicosity. This is a situation quite different in the single unit. That, remember, we said that because of the presence of the gap junctions and because these cells we just saw, right, some of them, they are self-excitable. So they have spontaneous electrical activity. So once one is activated, it will lead to the raise of calcium, and then all of this will be propagated to the neighboring cells, right? And then you have here, as is illustrated here, the innervation. The role of the innervation is mostly by modulation of the contraction that it was generated by a different mechanism. So I have here the micrograph for this single unit configuration, and you can see now the axons, and you can see that the varicosities, as you can identify here, they are more spaced. Okay, and this is the reason. I want to, okay, but when we look at the single unit, this resembles more to the cardiac muscle in terms like they are electrically coupled. So the electrical activity will be propagated between the tissue. And I have here again some examples of the muscles that will experience this type of activity, mostly rhythmical activity. We have the GI, the reproductive, urinary tracts, and also small blood vessels that will contract rhythmically. Okay. In the case for the multi-unit, this will resemble more to the skeletal muscle in terms that they are not electrical coupling and they are more influenced by the neuronal regulation. So as examples, we have the iris, vas deferens, the hair follicles, and even the larger blood vessels that, of course, they experience this tone, right? So I have one example, I always like to mention it. If we think about the uterus, the smooth muscle cells in uterus, normally you will find the smooth muscle cells in this configuration as a multi-unit. But once the uterus gets pregnant, then with the time, the smooth muscle cells will switch to this, from this multi-unit, they will switch to the single unit configuration. Why do you think they will do that? Because labor, right? So they need to get ready for labor. So then what you are expecting is that now they need to contract more uniformly, right? So in order to contract uniformly, they need to have these gap junctions, and then that's how the uterus will transition from the multi-unit to the single unit configuration, and then it will go back to the multi-unit once after some, I don't know, weeks from months from labor, right? So the uterus, for instance, is not innervated, and it will be controlled. These contractions will be under the influence of circulating hormones or even drugs, right? Remember, for instance, oxytocin, right, will be the hormone that will be circulating and affecting the contractile activity of the smooth muscle cells in uterus. So as is illustrated here, the hormones, circulating hormones and drugs, they can also affect the contraction of these cells. So here, I have, as I said, there are many hormones or neurotransmitters that modulate a smooth muscle cell. You saw already some of them in the previous lecture, so it's impossible to mention those, but here I have some examples that, for instance, adrenergic stimulation through the different types of receptors, you can get the smooth muscle cells either to decrease its motility or increase its contraction, right? For instance, in this case, activation of alpha adrenergic 2 or 1, it will lead to vasoconstriction through two different mechanisms, right? As is illustrated here, and I think that you already know this material, and it's the same for the parasympathetic stimulation, right? We have the acetylcholine that, depending on where are the smooth muscle cells, they will respond by contraction or by relaxation. Okay, here I have listed other factors that will influence contractions, another type of hormones, as I mentioned before, oxytocin, vasopressin, histamine. But we have also mechanical stretch that will usually open mechanosensitive ion channels that will lead to membrane depolarization. We have also local metabolites and drugs. In terms of drugs also, I think that you review and you know that some calcium antagonists or potassium channel openers will result in relaxation of the smooth muscle. And also we have that through this pathway, the nitric oxide cyclic GMP stimulation, the nitric oxide will relax the smooth muscle in a paracrine manner. And I have here in a dynamic cartoon how the nitric oxide do that. So I like to put this almost at the end, because then we'll put all the concepts together. So we have here the varicosity that we mentioned that contains the neurotransmitter, right? But in the varicosity also we got the activation of the nitric oxide synthase that will lead to production of nitric oxide, right? This NO will, this will diffuse out of the varicosity and reach the smooth muscle cell, as is illustrated here, and then this will bind to activate the guanylyl cyclase. This will lead to the production of cyclic GMP, and this will activate the PKG. And remember what will result in the activation of PKG? Relaxation of the smooth muscle, right? Because remember we said that PKG will phosphorylate the myosin light chain kinase, and this will result in relaxation. But at the same time, it could be that once this varicosity releases the neurotransmitter, in this case the ACH, this will come and reach the endothelial cells in the capillary lumen, and this will bind to the receptor that, because as we said before, and you know already, that through a G protein mechanism will result in the production of IP3. This IP3 activates the IP3 receptors with the release of calcium, then calcium rises and then activate the nose, and this will end with the production of nitric oxide that diffuses and also reach the smooth muscle cell. So here I have the temporal course of these actions, the ACH and the NO, that produces a rapid phase of relaxation. See? Questions? No? Okay, so I have in the next slide very cool videos that I think for you that you make an extra effort to come to the class. I guess that you are probably, this is going to be the only time maybe that you are going to witness the smooth muscle cell in action. So what I have here is, in the background, I have skeletal muscle cells. Probably you can distinguish with their vascularization. Probably you can see the vessels, right? So this muscle is from a mouse gluteus maximus muscle. So that means that this muscle, the activity of the smooth muscle cells, they were obtained in vivo. So actually the mouse is alive, is of course sedated, and thanks to this technique, intravital video microscopy, we can witness or we can monitor the arteriolar response to the topical application of some agents. In this case, this muscle were topically applied the sodium nitroprusside. So this is a NO donor. So what do you think is going to happen to the smooth muscle cells? It will contract, they will contract, or they will relax. Raise your hands that you think that they will contract. That they will relax? Okay, so let's see what happened. See, that movement is because the mice is breathing, okay? Isn't that cool? So now let's see. So what will happen if now we challenge the same smooth muscle cells, but now with norepinephrine? What do you think is going to happen? Relaxation or contraction? Contraction. Let's see. What? This is amazing. So you witness in vivo the pharmacomechanical coupling. Okay? So we are almost ready to go. So by now we just review the physiology of the cardiac, skeletal, and then the smooth muscles. So then we finish my blog. So let's now do our review of a smooth muscle. See? So in a smooth muscle, calcium binds to the thin filaments to enable contraction. True or false? False, right? Calcium drives phosphorylation of the thick filament, right? Because it binds to calmodulin. Okay, so the latch state permits long sustained contractions without fatigue. True or false? True. It's unique to the smooth muscle. Okay, what enzyme must act for a smooth muscle to relax? Remember the myosin light chain phosphatase, right? That is activated when the calcium concentration decreases. Okay, list two differences between single and multi-unit smooth muscles. Cell-to-cell electrical coupling because the presence of gap junctions, and remember they can initiate their own contractions. So what is called when contraction is initiated by a chemical or a hormone? Pharmacomechanical, we just witnessed that. Okay, and the last question. A smooth muscle is regulated by membrane potential variations. True or false? True, right? So sometimes can either initiate or modulate contraction. So we are done. So thank you. So remember, any questions, please email me, okay? Thanks for coming. Let me just eject my. Do you think it's this one? I don't know what it says. No, right? This is different. No, it's different.
Okay, I'm just gonna stop this actually.